Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're understanding the terms biodiversity and endemism. You'll also look at how biodiversity can be measured within a habitat using species richness and within a species using genetic diversity by calculating the heterozygosity index. You'll also understand how biodiversity can be compared in different habitats using a formula to calculate an index of diversity. So biodiversity is the variety of life in a particular area, and it includes diversity at three biological levels: the ecosystem diversity, species diversity, and genetic diversity. And high biodiversity is generally increases ecosystem stability and resilience, making it very important. So, a species is considered endemic if it is found only in one specific geographical area and not naturally found anywhere else in the world. And examples include lemurs in Madagascar and plant species on isolated islands, and you also have pandas in China. So, endemic species are at high risk of extinction because they have a small population size, restricted habitats, and limited genetic diversity. So, species richness is the number of different species present in a habitat. Again, another important slide. It is a simple measure of biodiversity. A habitat with more species has greater species richness. Species richness does not consider abundance of each species. So, it does not look at the numbers of an individual species. It just looks at how many species you have in a certain habitat. So, two habitats may have the same species richness but very different biodiversity. So genetic diversity refers to the range of alleles within a population. So high genetic diversity allows population to adapt to environmental change, resist disease, and reduce extinction risk. So the heterozygosity index is a measure of genetic diversity within a population. And this is given by the following formula. So the number of heterozygotes over the total number of individuals in the population gives you the heterozygosity index. So higher the H value leads to higher the genetic diversity, and lower the H value leads to lower genetic diversity and increased extinction risk. Alone is insufficient. So biodiversity is compared using an index of diversity, which accounts for two factors: the number of species and also the relative abundance of each species. And you can find the index of diversity using the following formula, where the capital N is the total number of organisms of all species, and the small letter n is the total number of organisms of each individual species. Professor SNAB today. Hope you found this video useful. If you have any questions, please let me know in the comments below, and I'll get back to them as soon as possible. And don't forget to like, share, and subscribe.